And greetings, everybody. Greetings, Wesley, Matt, and Patrick Alexander. I'm just trying to get all my buttons squared away here. So I have both me live and then my backup computer here. Just in case I have a little bit of a technical problem, the stream will continue. Let me just adjust my volume. And I'm also going to check the oh, Matt said, okay. Well, just to explain a little bit of yesterday's episode, it is important I have contact with people that have other viewpoints. And also, there are probably some subtle things that are kind of missed that I couldn't explain, like right in the middle of the stream, as far as teaching young Earth creationism and equipping. The goal really is, you know, they were framing it one way, what the discussion was about, and really it is to take people like Jonathan Guzman, who's like 17 years old, thinking about going to college. He's a young Earth creationist, and equipping him to face an environment that is very hostile. He'll face arguments that are not quite as, you know, they're not quite as easy as it's made out to be by other creationists. And then also there's some things in creationism, the arguments are actually not very good. or they're good, but they have weaknesses, they have Achilles heels, and I'm trying to shore those up. So these are very subtle rhetorical things that I've seen over 20 years of debate to avoid. So when I said, like, don't argue that ID is science, it's a rabbit trail. You're going to spend all this time, and you're not going in a discussion, and you're not going to be able to put complexity on the board because you're talking about metaphysical stuff or definitional stuff. The other side loves to get us in definitional arguments. The other thing is the origin of species. Discussions are made at the species level. It's just too difficult, too many variables, emphasizing going to the molecular level. The other part is irreducible complexity. I said it's not an argument, it's a heuristic. Go back to Darwin's original origin of species, and that outlines what you really need. So when Dr. Dan was having Michael Behe as a guest, they spent 45 minutes talking about the definition and applicability of irreducible complexity, and Dr. Dan would throw out all these systems. And I've seen it in debates where Dr. Dan will throw out a system he'll claim is irreducibly complex. It's in his field of specialty. And he'll say, well, I got to see this evolve. What do you do then? So I've said, don't use that argument. Don't use it as an argument. Use irreducible complexity as maybe a heuristic to find things that are unwinnable, undarwinable, unevolvable. And I'll cover that a little bit today. So it was good to engage the critics. Many creationists are so obsessed with the human-chimp common ancestry, and they don't argue it well. And I said, you argue the youth of the earth. It's a done deal if the earth is only 10,000 years old, or the fossil record's young, or life is young. Don't get into the genetics of this or paleontology. You have experts like primatologists like Erica. She's going to tear you to shreds. And I just see creationists go down these rabbit holes. I've been in these debates for 20 years. So, and it was really heartbreaking when Cindy Lincoln had her science tour and Dapper Dino just ripped it to shreds. And I was like, you know, this is exactly what you don't want to teach the kids. And I'm very, very concerned. So that was the whole thing about teaching young earth creationism and intelligent design responsibly. It's also effectively, so that they will be equipped to, better equipped. I guess we're never fully equipped. Better equipped to go to be in an environment, if they're going to go off to be scientists and medical doctors and professors, they don't get shredded to pieces when they go off to university, at least as far as the arguments go. So so thank you for the feedback, Mats. We're going to have more of those. Part of this also is I'm trying to write books, materials and articles to equip creationists to argue better. They have I believe they have the facts on their side, but they're not quite prosecuting. They're a little bit weak on the rhetoric side. And there are lots of rhetorical maneuvers where the other side will use equivocations and hasty generalizations, and a list of 60 or so logical fallacies. The most common is equivocation, and then hasty generalizations. And I just see it all the time. And sometimes I'm not that equipped. Oh, someone was asking about Emily Reeves. Okay, so I was Googling to find a picture of Emily Reeves. One of the hits you'll get is a different Emily Reeves. Emily Reeves is one of the ladies from the Discovery Institute. She's Dr. Emily Reeves, PhD biochemist. And there's also a professor, Greg Reeves, not at all related. But I do also know, oddly enough, Greg Reeves. So, so today I'm preparing a dress rehearsal for my presentation to the Discovery Institute group, engineering group. One of my co-authors actually, in a paper that we both published, Joe DeWeese, he's a Discovery Institute fellow. And so I'll showcase some of that work. So as I was, tomorrow it's going to be semi-formal presentation. This isn't like going to be a big deal. This is going to be in their engineering group. And this is just going to be a little bit of a free, somewhat free discussion. So this isn't going to be the most polished thing. It's meant to be, it's actually, we call it the engineering working group sometimes informally. That means we're going to bounce around ideas. And, they said, we'll roll up our sleeves, so to speak. We're not out there to have the most polished presentations. So what I'm going to do today is kind of run through my 150 or so slides and just see how efficiently I can get through this. I'm going to pare it down. I'm going to try to pare it down to after this round. So we'll just see how effectively I could present this. There may be some technical problems because generally, whenever I have PowerPoint and StreamYard running on my main computer, it doesn't mix. I might have to, let me see, I'm going to go to my backup computer. I might have to be trying to run some of the videos on my backup computer. So if you'll bear with me, this is just a practice run. It's going to be pretty clunky. And I would get I'm getting a problem with my backup computer already. It's saying couldn't verify my subscription, which means I have to reinstall it on my backup computer. And I have to try to run this, see if I can run, let me try to run one video, see if it'll allow me to do that. No, it's not going to let me. Oh, Tony Maurice and Mark O'Connell. Okay, great to see you guys. All right, I have my first technical glitch already. So I'm going to have to go to plan B and yank. I have to get a thumb drive on one computer because I don't have my computer's network and download some videos real fast to the thumb drive to my. So if you'll, again, thank you all for your forbearance, and I apologize for my totally messing this up. So that's one glitch I already know I need to deal with. So let me download at least one video. Let me transfer one video, and then while I'm running it, I'm going to transfer the rest. So that way this will proceed a little smoother. Okay, get this out. And so okay, so I think I'm a little bit better set up, not quite. So let me bring up my PowerPoint now. Jamie, greetings. And let me if someone's a mod, if you could start blocking some of these spam bots, that would be great. Oh, by the way, I think Matt's, I made you a moderator, Matt's. Congratulations. And greetings, Jamie Russells. So tomorrow, I'm going to be giving a talk on protein probabilities. That's the title, Geometric Connectivity, NLS, which is nuclear localization signals, PTM, Post-Translational Modifications, Translocation. LLN is law of large numbers. So that's my presentation tomorrow for the Discovery Institute Research Group, January 29th, 2022. Some of you already know who I am. I'm going to just point out I have a connection to Casey Luskin at the Dr. Casey Luskin at the Discovery Institute. We are both featured in the scientific journal Nature in 2005, the April 27th, 28th. So that's kind of my my roots to the Discovery Institute. And as some of you know, I was at the Dallas conference last week. My presentation tomorrow was scheduled like 2 months ago, so it just happened to overlap that we're so close that the people I saw at the Discovery Institute I'll see tomorrow, many of them. So the engineering group is run by Brian Miller, Steve Laufman, Emily Reeves, and Casey Luskin will be present, probably some of the other Discovery Institute fellows and staff will be there, plus interested parties. I heard the group is between 15 to 30 people. And that's always fun. This is how I have the chance to actually even make presentations to faculty around the nation that are interested in the topic of intelligent design. So one of the ways to introduce the topic, there's been Michael Behe's book, Darwin Devolves, and this whole thing about fitness. Oh, and someone asked, will it be uploaded to YouTube? Actually, that's a private meeting, but you're hearing it's essentially the presentation now. In fact, you'll get more in this presentation because I have to chop this down. Oh, and LPP is here. Oh my goodness. So LPP, just to update you, I'm making a presentation to the Discovery Institute tomorrow. This is just a dry rehearsal, kind of a, not even, it's meant to be a dress rehearsal, but it's not quite that polished. I was at the Dallas Discovery Institute conference and also the ICR last week in Dallas, Texas. And I'm sorry I missed you, John Maddox, as I was passing through Nashville. So one of the things I'll point out is Darwin Devolves, Michael Behe's book. This paper by Lenski himself highlights the problem. It says mutator genomes decay despite sustained fitness gains in a long-term experiment with bacteria. This, I get flack from the other side and I respect them, but this just points out what I've been saying is the ludicrous definition of fitness in evolutionary biology. The genome can be getting trashed, and yet fitness is increasing. That's ridiculous. I mean, what's the thing measuring as far as, I mean, it's like, oh, you have more of this, you have more of these alleles. And it's like, duh, or lack thereof. So as this genome's getting trashed and possibly now in some of the lineages going to extinction, so what if some of the individuals are being more reproductively abundant? Because the problem for evolution is not making more babies. The problem is evolving complexity and new organs. And this is a disconnect. And I couldn't have picked a better title to highlight the problem with the evolutionary definition of fitness. The genome decays despite sustained fitness gains, because fitness in evolutionary biology means making more babies. And if that means that the stupider and dumber make more babies and their genomes decay, well, that's an increase of fitness in the evolutionary world. So if there is an abundance of obesity and diabetes and Tay-Sachs disease and sickle cell anemia, if it leads to some reproductive success, it's considered fit. And there was a book by an evolutionary promoter to that effect called Survival of the Sickest. And so the problem is that beneficial mutations in the Darwinian sense are they're bad. Okay, they're loss of function. Oh, and my goodness, I need to acknowledge this. I kind of got stopped in my tracks. God bless you, Alyosha. And I will. This motivates me to try to be of service to the community here. So thank you. Thank you very much. So again, why am I highlighting this? Well, we got our book out. We got our chapter in the Handbook of Mathematics and the Arts and Sciences by Springer Nature, which is a respectable publisher in the scientific world. We got the invitation to write the chapter because there was a peer-reviewed paper that got the attention of in the Journal of Mathematical Biology that set all records for the number of downloads, and it got the attention of the editor of this publication, and we were just given an invitation to speak our mind. And so we restated a lot of the paper and added some extras. Now, just so you know, I don't get a dime for this book. So thank you, Alyosha, for your donation. Because if you guys buy this book, it's the publisher who gets this. We only get benefits for contributing to this book by getting a free copy of the book electronically. Okay, so that was the deal that was worked out so that we could be compensated, but we don't actually get any money. And for the diehards out there, if you, oh, by the way, this is the really funny part on the Springer advertisement. They said this was edited and written by leading scientists. So I got a chuckle out of that. They're calling me a leading scientist. That's just kind of funny. You can get it at Walmart for $819, which is kind of cute. So this was our publication. This is our chapter in that book, in that very expensive book. We were able to submit this because we just followed the tactic of reviewing a lot of what evolutionary biologists themselves have said about their own field, such as this paper written by an evolutionary biologist, and he's highlighting what's wrong with evolutionary biology. So there's really no reason they should retract this paper. They might because this chapter, they might because we're hostile to evolution, but in our chapter, we just highlighted what evolutionary biologists themselves. So there's nothing we said that was outside of the mainstream. And again, this is some of the stuff in our paper that was also in the Journal of Mathematical Biology. William Basner's co-author, John Sanford was my co-author, and again, some more of the math. And we had Ola Hosger, who's trained like 14 PhDs, award-winning mathematician in Sweden, University of Stockholm, and he has like 14 PhD students. So I had great co-authors on that paper. One of the things that we covered in that paper was I put forward, I said, I think this is, we may not have another shot at this, guys. This is a good time to talk about the evolutionary definition of fitness and why it is illogical. That was not new. People like Richard Lewontin and others realized it was a vacuous concept. R.H. Brady is another one, but Lewontin was the big name. He said this is a nothing concept. I mean, those weren't literally his words. If you go to our chapter and look in the bibliography, you'll see a reference to Lewontin's criticism in the 2003 Santa Fe Winter Bulletin. So I'm just going to move on, otherwise I'm going to run out of time. Let's see, we started about 11:20. Okay, so let me just keep plowing forward here. So how do we define fitness alternatively? I have suggested we use the idea of biophysics. We know what is the limit of, we know what is the, are the limits of physics in building a system. And Princeton physicist, biophysicist William Bialek, or I call him Bialek, he made a presentation and he's done work on showing that life is more perfect than we imagine. That is, if you're trying to build systems to the absolute limit of physics, biology does that in many dimensions, from optics to sensing photons, to sensing individual molecules, to counting individual molecules, to sensing magnetic and electric fields. It's at the limit. In fact, it's so good we cannot build. Right now, with our present technology, if we were to implement these things at the molecular level, we would not be able to build them as well as we find them in biology. So hence, I put forward, and thankfully my co-authors agreed, the idea of defining adaptation of a system to its environment in terms, in engineering terms, and in physics terms, rather than the vacuous notion in evolutionary biology, you're just counting how many babies it makes, which doesn't prove anything. It doesn't prove a system's going to evolve complexity. It doesn't really show how well adapted it is to the environment. It only shows that maybe one allele's a little bit better adapted than something else. But what biophysics demonstrates is when a system is adapted to the environment, to the physical environment at the absolute limit, it tells whether it really is or is not. Those are far better metrics than the definition of fitness in evolutionary biology. And that was the point, that was one of the points we put forward in our chapter. So now that I'm there, I'm going to take a risk here and let me try to play a video. Of the Potassium Ion Channel.
The bacterial potassium channel is a multi-pass transmembrane protein in the plasma membrane. It is built from 4 identical subunits that are arranged symmetrically. A pore in the center of the protein allows selective passage of potassium ions across the membrane. 4 rigid protein loops, one contributed by each subunit, form a selectivity filter at the narrowest part of the pore. This structure is responsible for the channel's high degree of selectivity for potassium ions over sodium ions. In this selectivity filter, carbonyl groups line the walls of the pore. These carbonyl groups are spaced precisely to interact with an unsolvated potassium ion, balancing the energy required to remove its hydration shell. Passage of a sodium ion through the channel is energetically unfavorable because the sodium is too small for optimal interaction with the carbonyl groups.
All right, I hope you enjoyed that. Let me just, I have to do a little cleanup here on the videos. And okay, this is why it's nice to have a co-host, and I don't have one right now. So thank you again for your forbearance. So let me point out some things. Now, normally when you drill a hole, you expect anything smaller than that hole to get through. At the molecular level, it works differently for the potassium ion channel. So remember, the potassium ion here, and you can see the size is 1.33 angstroms. The hole is fit perfectly to allow this to go through. If it's too big, it's obvious it's not going to go through. The amazing thing is that if it's too small, and I know this sounds counterintuitive, if it's too small, it's not going to go through either. This is some really difficult. Okay. Someone said there was no audio. Let me try playing it again. I'll try playing it again. Did anyone else have audio issues?
The bacterial potassium channel is a multi-pass transmembrane protein in the plasma membrane. It is built from 4 identical subunits that are arranged symmetrically. A pore in the center of the protein allows selective passage of potassium ions across the membrane. 4 rigid protein loops, one contributed by each subunit, form a selectivity filter at the narrowest part of the pore. This structure is responsible for the channel's high degree of selectivity for potassium ions over sodium ions. In this selectivity filter, carbonyl groups line the walls of the pore. These carbonyl groups are spaced precisely to interact with an unsolvated potassium ion, balancing the energy required to remove its hydration shell. Passage of a sodium ion through the channel is energetically unfavorable because the sodium is too small for optimal interaction with the carbonyl groups.
Well, anyway, for those of you who did hear it, that's great. And we just do a little more cleanup here. So I was mentioning that the potassium ion channel has to be the right size. And in fact, in the video it said perfect fit, perfect fit. So what does that mean? If you just take random mutations on this thing, you're not going to make it any better for sure, because it has to be perfect already for it to work. I mean, we're talking about precision in the angstrom, sub-angstrom range. That means you could see the radius of the potassium ion here and the sodium ion. This is fractions of the size of the atom. This is how precise this has to be. In the man-made world, it's very hard to machine things with that level of precision. So the idea of fitness, at least in this case, is physically in terms of a geometric fit. And I often, here's a picture of a nut for like a nut and bolt on the left. On the right is amazingly a different look at the potassium ion channel. I believe that's potassium. That looks like a voltage gated channel. I edited it just a little bit to emphasize really the square symmetry it has. And it's very precise. It's more precise than the hole drilled in those nuts there. If you didn't have potassium ion channels, your nervous system wouldn't work, so you'd likely be dead. That's just how critical this is. And again, there's a picture of it. As I said, it says perfect fit, perfect fit. This is not defined, we don't define that this is a perfect fit in terms of reproductive success. It's described in terms of physics, which is a better description of adaptation. And don't even think about adaptation. We're just thinking about the ability of this machine to operate at the limits of physics. Here's another picture of the potassium ion channel. So just so you know, this, when you have these ribbony diagrams, that's a Richardson ribbon rendering of the protein, the potassium ion channel. This one is the ball and stick, I believe, model. Just different renderings. Oh. All right. Now, let me point out something. If I just gave a requirement specification to say, construct a filter that'll only allow potassium ions to pass through. If I gave that to a chemist and he didn't know these existed, do you think he could be able to construct it? Seriously, I've asked chemists about systems like this. They said, no way. You could take the very best. I talked to a physicist who worked at a Nobel Prize winning lab, and we were talking about these systems. He said, you couldn't take the best scientists in the world and succeed. The problem is that difficult. So we might wonder, well, we're able to send people to the moon and build space probes and do all these things. It's a different story when you try to engineer at the nanomolecular level. Building big macroscopic objects is relatively easy compared to building things at the very, very, very small scale, where you're dealing with atoms and then trying to, like effect, make sure you're using electrons that have only a certain spin, you know, the chiral spin selectivity. I mean, we see a lot of that in biology. We don't have the technology to build systems like that. So in, you know, the way that we do things in the macro world is we drill holes. Hang on, let me put this back up. Sorry. If we want to make a hole, you know, quote unquote, a channel, we'll drill holes. But in the molecular, at the molecular level, we create these holes or channels by spelling. So we have to figure out a way to spell something, and when we spell it, it's going to make something like the potassium channel. This is not trivial to do. This is a very difficult problem that we don't know how to solve. So spelling defines size and shape and function. So if I said make something that's shaped like this, do you think you could do that? If you didn't know about the potassium ion channel to begin with. And a lot of scientists will say, no, that's not possible. So we may think because we have genetic engineers like John Sanford that we figured out how to do this, and I can tell you we haven't. When we do genetic engineering, we're doing a lot of transplanting of what's already in other biological organisms. We're not able, you know, we're not able to build stuff like a potassium ion channel from scratch. We have to borrow parts. Okay, so when we do genetic engineering, it's nothing like really building it from scratch. So that's my boss, Dr. Sanford. He would know because he was a famous genetic engineer. So let me just point out a little bit about something in the amino acids. We have, there are the 20 amino acids, And you can see the kind of the colored regions of each amino acids. That's called the R group. That's the part that's different, that distinguishes one amino acid from another. And I can depict what the R group has to look, how it can be oriented. There's an orientation of the amino acid that will make the R group point one way or the other. And in this depiction here, you have two proteins that have charges distributed on them. The R groups may have charges associated. They may be positive or negative. And so when you want to have two proteins connect together, you had the minus charges on one protein and the plus charges on another, and they have to be shaped right, and then they will connect. Hang on, let me toss someone here. And oh, we have someone, a first-time watcher. Thank you. So it's not exactly easy to get the charges oriented in the right way. We could go into alpha helices and beta turns. This is not trivial. That's the first thing. And you have to have foresight that this protein is going to connect to another protein or some other thing to get the charge distribution right. So you have to get the connection points right. So these are where you start to see the improbabilities of forming proteins that are functional within a context. Here are the list of the amino acids and some of their properties in a Venn diagram. So the potassium ion channel, and you can see where it says R. The R groups have to be pointed to certain directions so that it can interact with the potassium ion as it's moving through. This is, again, not trivial. You have to have foresight to be able to build something like this. Again, this whole thing with the R groups, this is a diagram that probably wasn't so successful, where I'm trying to show that the positive and negative charges have to be oriented the right way. Another way of conceiving of this is just like this little 3D puzzle for the child. You can put it together. Every part has to fit. And the proteins are very much the same way. This is a picture of a protein here, and you can see all the colors. The parts of the protein have to connect to each other, and you can see it right here. This is the EZH2 complex, and I'll point out where that connects. So it really does remind me of this kid's toy. So fitness in the biological sense is not really well expressed with the evolutionary definition of just making more babies that live to adulthood to reproduce. It's better to talk about it in terms of the geometric connectivity and action, et cetera, et cetera. I mean, there are better ways to characterize this than just kind of these static diagrams, but this is a start. And if we define fitness kind of with this feeling of getting the geometry right, it becomes clear why most mutations are damaging. Do evolutionary biologists consider this sort of thing when inferring relationships? I think they don't do it enough, if they do it at all, honestly. Sorry to pick on one of our friends, Andrew Cumming. He doesn't seem to appreciate this. I try to tell him, and he just sticks to those phylogenetic reconstructions. It's just, I don't know. It's like, Andrew, how can you look at these considerations and think your phylogenetic reconstructions explain all this connectivity and shaping? So let's move on. And I don't know how I'm going to fit all this in my talk tomorrow, but that EZH2 molecule here, not only do the internal parts have to connect, as you're seeing right here, they connect with other things. So here's the EZH2 right here, and all these other things it connects to. It's just unbelievable. it's connecting to the DNA here, which is the wires, this complex, the polycomb repression complex 2, and all these other proteins that are just as complicated. And then there's a non-coding link RNA somewhere here. This is just insanely complicated. Insanely complicated. And that's another story. I'd love to talk more about this. So just kind of as a rough way, just think also about fit, the adaptability has to be, the parts have to be geometrically sensible to be able to connect to each other. I mean, if you're putting together a car, all the parts have to fit. All the parts have to fit. So this is how something can be unadapted to the environment if it has broken parts. So, that's one constraint, but there are more constraints. We have two major architectures, the prokaryotic and eukaryotic architecture. This is a big problem for evolutionary biology. How we can evolve a prokaryote like this into eukaryote that has all these membrane-bound organelles. Because you have more locations in the cell, in the eukaryote, that have to have special security codes, so to speak, to get through. It's more like addresses. So the protein needs to have an address on it, kind of the address typed on the letter, so to speak. And then the postal delivery guy is going to move that package to the right location in the cell. And there are all these other compartments it has to go through. Yeah, good question. Thank you. So now I'm going to show a video. I'm going to see if I could do this successfully. So one of the ways that I'm going to show a video that shows how when we have the DNA in here, it gets transcribed into RNA. The RNA will wander to one of the membranes, and then we're going to see the process. We're going to see the translocation process, or parts of it. It's probably more complicated than what we're going to see in this video. But the protein, not only does it have to be shaped right, it has to have translocation codes in it if it is the sort of protein that needs a translocation code. So let me try to share a video on that. All right.
The term secretion is used to describe the movement of a protein out of the cytoplasm. A gram-negative cell has a number of destinations for secreted proteins. Secretion systems move proteins into the inner membrane, the periplasm, the outer membrane, or outside the cell into the surrounding environment. Special export systems are required to move hydrophilic proteins through the hydrophobic membrane barriers. Let's examine how proteins are delivered to the inner membrane. As a ribosome translates a protein destined for the inner membrane, the first part of the protein to emerge from the ribosome tunnel is a very hydrophobic region called a signal sequence. A cytoplasmic structure called a signal recognition particle, or SRP, quickly binds to the signal sequence. SRP, which is a complex of RNA and protein, essentially paralyzes the ribosome. SRP delivers the stalled ribosome and its nascent protein to the cell membrane, where SRP binds to a membrane protein called FTSY. Although some proteins spontaneously insert into the membrane, most use a general secretion complex composed of 3 proteins, collectively called the sec-YEG translocon, embedded in the cell membrane. In this process, SRP dissociates from the complex. prompting FTSY to deliver the signal sequence to the translocon. The newly made membrane protein may have many membrane-spanning regions. These hydrophobic regions, 20 to 25 amino acids in length, are important because they're compatible with the hydrophobicity of the membrane itself. The hydrophobic regions move laterally out of the complex and into the membrane. The looping may occur many times, considering that many transmembrane proteins have as many as 12 such membrane-spanning regions, which weave back and forth across the membrane.
Sorry, guys. No audio. My bad. My bad. So we saw, what I was saying is, we saw that you saw this translocation process where it's stitching it into the membrane. In the membrane here in a eukaryotic cell is like on the outer part. The potassium ion channel is one such protein that needs to be stitched in. So not only does the potassium ion channel have to be shaped right, it has to have all these codes that enable its construction and incorporation and integration into the membrane. So remember, this the DNA is in here, it gets transcribed, it comes out here, it has to be shuttled to the membrane, it can't just go anywhere, and then when it gets there, it has to be stitched in. And it's really bad if you're locating the proteins in the wrong location in the cell and stitching it where it's not supposed to be. I mean, you know, these phylogenetic trees that the evolutionary biologists keep drawing, it's like that's not an explanation for how selection will construct a system of this level of complexity. So now let me just show another video that will show the translocation process for proteins that end up in the endoplasmic reticulum. It's a similar idea, which will even get more details, and it would be applicable to any sort of, it would be extensible probably to a lot of stitching and weaving of proteins that are in any of the membrane-bound organelles. So let me get that started.
Endoplasmic reticulum, or ER, is the most extensive membrane system in eukaryotic cells. The endoplasmic reticulum, or ER, is the most extensive membrane system in eukaryotic cells. Proteins transported to the Golgi apparatus, endosomes, lysosomes, and the cell surface all must first enter the ER from the cytosol. As an mRNA molecule is translated into a protein, many ribosomes bind to it, forming a polyribosome. There are two separate populations of polyribosomes in the cytosol that share the same pool of ribosomal subunits. Free ribosomes are unattached to any membrane. Membrane-bound ribosomes become riveted to the ER membrane and translate proteins that are translocated into the ER. These membrane-bound ribosomes coat the surface of the ER, creating regions called rough endoplasmic reticulum. Two kinds of proteins are moved from the cytosol to the ER. Water-soluble proteins completely cross the ER membrane and are released into the lumen, while transmembrane proteins only partially cross the ER and become embedded in the membrane. All these proteins are directed to the ER by a signal sequence of small hydrophobic amino acids. The signal sequence is guided to the ER membrane with a signal recognition particle, or SRP, which binds the ER signal sequence in the new protein as it emerges from the ribosome. Protein synthesis then slows down until the SRP ribosome complex binds to an SRP receptor in the ER membrane. The SRP is then released, passing the ribosome to a protein translocation channel in the ER membrane. Thus, the SRP and SRP receptor function as molecular matchmakers, connecting ribosomes that are synthesizing proteins containing ER signal sequences to available ER translocation channels. In addition to directing proteins to the ER, the signal sequence functions to open the translocation channel. The signal peptide remains bound to the channel, while the rest of the protein chain is threaded through the membrane as a large loop. Once the protein has passed through the membrane, it is released into the ER lumen, after the signal sequence has been cleaved off by a signal peptidase located on the luminal side of the ER membrane. The signal peptide is then released from the translocation channel into the membrane and rapidly degraded. It is thought that a protein serving as a plug then binds from the ER lumen to close the inactive channel. But not all proteins that enter the ER are released into the ER lumen. Some remain embedded in the ER membrane as transmembrane proteins. For clarity's sake, the membrane-bound ribosome will be omitted to illustrate the translocation of transmembrane proteins into the ER membrane. In the simplest case, that of a transmembrane protein with a single membrane-spanning segment, the N-terminal signal sequence initiates translocation, just as for a soluble protein. But the transfer process is halted by an additional sequence of hydrophobic amino acids, a stop transfer sequence, further in the polypeptide chain. The stop transfer sequence is released laterally from the translocation channel and drifts into the plane of the lipid bilayer, where it forms a membrane-spanning segment that anchors the protein in the membrane. As a result, the translocated protein ends up as a transmembrane protein inserted in the membrane with a defined orientation. In some transmembrane proteins, an internal signal sequence is used to start the protein transfer, which continues until a stop transfer sequence is reached. The two hydrophobic sequences are then released into the bilayer, where they remain anchored. In complex multipass proteins, in which many hydrophobic regions span the bilayer, additional pairs of stop and start sequences come into play. One sequence reinitiates translocation further down the polypeptide chain, and the other stops translocation and causes polypeptide release, and so on, for subsequent starts and stops. Thus, multipass membrane proteins are stitched into the lipid bilayer as they are being synthesized, by a mechanism resembling a sewing machine.
Okay, this time I remembered to turn on my microphone. So you could see the protein has to be formatted with these stop transfer sequences and all these other things to get it properly processed by the machinery that's incorporating it. There are lots of these transmembrane proteins. One of them I showed is Potassium Ion Channel. There's like the Sodium Ion Channel, and it has to be stitched in the membrane that way. How much foresight would it take to write all these codes, these multiple codes, into that protein and then also get it to fold right? That's why I'm just giving a feel for how difficult it would be for a protein engineer to, if you were given these specifications, build this from scratch. See that it's compatible with all the hardware in the cell. This is not trivial. So let me keep going. Let's see, what's my next topic? Oh, there's a Wikipedia entry on this, on target peptides, if you want to read some of this. It's just great that we have videos, because then you kind of see just the power of it without all the technical jargon. You could see just the complexity, and appreciate it. So we have lots of we have lots of locations in the cell, and we already went through these videos. This is beginning, now we can begin to understand why most mutations are going to be damaging. And in the case of some of the proteins we covered, it's like, how can you actually get them to improve? They're already, they already fit perfectly. You might be able to like add extra functionality, like in the case of the potassium ion channel, you might have you might have one that's always open, and then some that will open and close based on the voltage, or in the case of other ion channels, you'll have the channels open and close because of the presence of a ligand or something else. So going back to this here, in the engineering sense, it's very hard to get a truly improved system. And in fact, this diagram here was based on the evolutionary definition of fitness, where fitness is just making more offspring. It's not even considering the mechanical things. If we consider the mechanical things, then even a lot of the beneficials are only beneficial because they're loss of function, not gain of it. And that was really the subject of our Springer Nature chapter. And it's actually echoed in some of the papers if you're really willing to read carefully, like where Lenski admits you can have fitness gain and you're just trashing the genome in a functional sense. So now let me show something you've seen me cover before, but for the engineering group meeting tomorrow, we're going to showcase this. One of the fellows at the Discovery Institute was my lead author, Joe DeWeese, and so we'll just show this.
Consider what happens as DNA unwinds during replication. As DNA unwinds, let's consider what happens as DNA unwinds during replication. As DNA unwinds, it acts like this rope when we pull apart its two strands. As you pull the strands apart, twisting tension builds up in the rest of the coiled portion. It is actually adding one twist to the remaining rope for each twist pulled out of it. At some point, you can't separate the strands anymore. The remaining rope is too tightly twisted. If you relax your tension on the rope, it will twist around itself in a supercoil. It is releasing tension. If you want to keep pulling the rope apart, you have to release the tension periodically, and one way to do this is to cut the rope and splice it back together. This problem has been best characterized in small circular DNAs. There are two methods of dealing with this problem in DNA. One cuts only one strand of the DNA double helix, and the other cuts both strands. Let's look at the first. Topoisomerase I enzymes cut a single strand of the double helix, pass the other strand through the cut, and reseal the break, relaxing the overwound molecule, which now has one fewer twist. Topoisomerase II enzymes do the same thing but with both strands of the double helix. Topoisomerase II cuts both strands of a double-stranded DNA and passes another double strand through the break and then reseals the break. So if a molecule of DNA is supercoiled, topoisomerase II can remove the supercoiling, 2 twists at a time, to yield a relaxed circle.
I'm going to show a different version of the topoisomerase. It'll add a little bit more, a different perspective, which I will talk about right afterward. So this video is done with clay. So how do we know that this is pretty important? It's the target, as you can see, of various cancer therapies. The way we can kill cells is you disrupt topoisomerase function. So this leads to a problem for evolutionary biology. I'm going to try to cover some of that. Let's see. We have, oops, let me clean this up. Just leave some of these videos hanging on there. I need to remove them. Okay. Let me do a little more cleanup. So Darwin said this. This is from chapter 6 in the section on organs of extreme perfection and complication. It says, if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down, but I can find no such case. Now, one of the problems with this is I've asked Darwinists, what would be a good enough demonstration? What would be enough to show you that this would not evolve by slight, numerous successive slight modifications? So, we could, how many experiments would we have to run on topoisomerase that if you just randomly mutate it and it breaks, how many experiments would we need to show that this won't evolve by slight successive modifications? How many proto-ancestors of the topoisomerase do we have to run this experiment on? And by the way, if topoisomerase is missing to begin with, the cell most likely will be dead. I don't know. There are things that are simple, like things like the phage virus, which isn't cellular life, that don't have to untangle its DNA, but I don't know about cellular life. But suffice to say, this is a problem, and I'll tell you why. So let me see if I have another diagram here. Okay, so the first thing is, I'll just show this is the spelling of the topoisomerase protein. It's only about 1,500 amino acids long. I've tried to color some of the what they call secondary structures, the alpha helices, the beta strands, the turns. It's neither here nor there, but let me point something out. You see this pair of scissors here? Scissors. Topoisomerase, as you can see, the topoisomerase 2 alpha and 2 beta are like a pair of scissors. So just like a pair of scissors, you have two parts that are identical, and when you connect them together, you get the scissors. Topoisomerase, to get a functioning topoisomerase, you have to have 2 copies of the protein, just like a pair of scissors, then they come together. And I'm highlighting here in the various colored letters, those are the connection points. And again, it has to be in the right place to make this a sensible pair of scissors. So going back to the Darwin quote, if you have a topoisomerase and all it can do is cut, you're going to shred the genome. Now there are things like nucleases that do cut, but they have to be, if they cut, there has to be something else that stitches it back together. So if you have a topoisomerase in isolation and all it can do is cut, and you don't, and there's no other means for stitching it back together, the genome's shredded, the creature's effectively dead, or its lineage is dead. So it's like good luck just having a cutter if you don't have a means of connecting. Likewise, if you just have something that connects and it doesn't cut, that's not going to work. Or if it connects and cuts but doesn't untangle before it reconnects, that's bad. If it doesn't know how to sense the tangles and locate, that's bad. And I put this forward, and they said, well, you have to have a demonstration. I was like, well, how much more of a demonstration do you need? I've given you one in principle. The other thing is, like, where's the burden of proof here? If evolutionary biologists are claiming this evolved, and they can't provide proof that it did, except in their imagination, that's not science. That's a statement of faith. So I think in light of what we've just seen, I don't think it looks really good for the Darwinian viewpoint for systems like this, because they can only invoke that it evolved as a statement of faith, or evolved by natural selection as a statement of faith. It's certainly not anything that can be demonstrated from physical theory, chemistry, what we know about cellular biology and statistics, you name it. It's only in their imagination. And their rather flimsy definition of fitness is not going to cut it to even provide an explanation. So, but that's not the end of the problems. Going back to the so we talked about the locating problem, the targeting problem, or the transportation problem. Let's just walk through what has to happen in for the topoisomerase to work. So for the transmembrane proteins like the potassium ion channel, you have the DNA becomes an mRNA, transcribed to mRNA. The mRNA gets here, and then the protein is expressed. It is translated by the ribosome, and it gets stitched into the membrane. Topoisomerase is different. Topoisomerase, the gene is in here, transcribes to RNA. By the way, the RNA has to know to get out, so there's an addressing problem there. The address is put in something I think is called in the poly A tail of the mRNA, but there's still an addressing problem. So it has to go out here. It'll get translated. The mRNA gets translated into a protein, the topoisomerase protein. The protein has to go back in here to untangle the DNA. So this is something of a chicken and egg paradox, because if you don't have the gene for the topoisomerase, you don't have a topoisomerase which can go in here and untangle the DNA. So without the DNA, you don't have the topoisomerase. Without the topoisomerase, you don't have a functional DNA. So how does the topoisomerase know how to get back in here? And some of you have seen me talk about the nuclear import-export, but since we have a newcomer here, we'll show him the video of the nuclear import-export process. And pay special attention to the word nuclear localization signal. I'm going to show you the spelling of that in topoisomerase. It's huge. It's a huge password. So let's see the nuclear. See if I could find it. I hope I have it. Protein translocation. Okay, great.
Endoplasmic reticulum, or ER, is the most extensive membrane system in eukaryotic cells.
Hang on, I have the wrong video. Let me get the right one. Oh. All the headaches with StreamYard. Well, that's on me. I wasn't quite prepared for tonight. So Let me find it. Nuclear import export. Here it is. Hang on. I'll get it relocated. All right. Let's all right. Let's go share video file. Let's try to do a refresh. It can't see it. Oh no. All right, let me try a different way to do this. Nuclear import export. I'm gonna try a different way, guys. Let's see if I could do this. Give me a moment. We'll try it a different way. Share system audio. Let me know if you get audio.
Molecules, ions, and macromolecules can travel between the nucleus and the nuclear pore complexes are the only channels through which small polar molecules, ions, and macromolecules can travel between the nucleus and the cytoplasm. By controlling this traffic, the nuclear pore complex plays a fundamental role in the physiology of all eukaryotic cells. Most proteins and RNAs are too large to move through the complex by passive diffusion and must move by an active process in which appropriate proteins and RNAs are recognized and selectively transported in a specific direction. Let's consider proteins imported from the cytoplasm to the nucleus. Only a subset of proteins can enter the nucleus, and they are allowed in by virtue of having specific amino acid sequences called nuclear localization signals. Nuclear localization signals typically consist of one or two short segments of basic amino acids. Nuclear localization signals are recognized by nuclear transport receptors called importans, which carry the cargo proteins into the nucleus. With its cargo attached, importin binds to specific nuclear pore proteins in the cytoplasmic filaments. By sequential binding to more interior nuclear pore proteins, the complex is translocated through the nuclear pore. At the nuclear side of the pore, the complex is disrupted by the binding of a protein called RAN to importin. RAN carries a molecule of GTP. This binding changes the conformation of importin, which then releases its cargo protein into the nucleus. The importin RAN complex is then re-exported through the nuclear pore. A protein in the cytoplasm called RAN gap, for GTPase activating protein, stimulates RAN to hydrolyze its GTP to GDP, an action that triggers RAN to release importin back into the cytoplasm. RAN plays a key role in protein import and export. Note that while RAN is bound to GDP, it cannot disrupt the binding of importin to a cargo protein. In an action that prevents the depletion of RAN from the nucleus, the RAN GDP formed in the cytoplasm is transported back to the nucleus by its own import receptor, a protein called NTF2. In the nucleus, another protein called RAN-GEF, for guanine nucleotide exchange factor, stimulates RAN-GDP to release its GDP and pick up GTP. In this form, RAN-GTP can disrupt the binding of importin and its cargo, triggering the release of the cargo in the nucleus. By the actions of RAN-GEF, found only in the nucleus, and RAN-GAP, found only in the cytoplasm, a steep gradient of RAN-GTP and RAN-GDP is maintained across the nuclear membrane, with RAN-GTP inside the nucleus and RAN-GDP in the cytoplasm. Similar to imported proteins, proteins are targeted for export from the nucleus by specific amino acid sequences called nuclear export signals. Nuclear export signals are recognized by receptors within the nucleus called exportins, which direct protein transport through the nuclear pore complex to the cytoplasm. RAN-GTP promotes the formation of stable complexes between exportins and their cargo proteins. Note that this same form of RAN does the opposite for importins and their cargoes. The effect of RAN-GTP binding on exportins dictates the movement of proteins containing nuclear export signals from the nucleus to the cytoplasm. Following transport to the cytosolic side of the nuclear envelope, GTP hydrolysis and release of RAN-GDP leads to dissociation of the cargo protein, which is released into the cytoplasm. Exportins are then recycled through the nuclear pore complex for reuse. The nuclear pore complexes are the only channels through which small polar molecules, ions,
Wasn't that cool, guys? So, in a debate with James Downard, he thought he had me when I was talking about eukaryotic evolution. Eukaryotes have machinery like this that prokaryotes don't. You could see that this is life critical to the cell. He thought he had me when he was going to argue about endosymbiosis theory shows eukaryotes could evolve. I showed this to him. He was just, I think I slapped him up the head so silly he didn't know what to say. He said something like, "Oh yeah, that's really interesting. We need to figure that out." I'm like, duh. This is a problem with eukaryotic evolution. Let me see if I could show another video. So you saw that nuclear pore complex that has, I'm going to play a silent video here. Maybe I'll try to narrate it while it's playing. Let me see if I can do that. Nuclear pore complex. So this is the nuclear pore complex, which we just saw. 456 proteins of 30 different types. So you remember the stitching, the membrane spanning proteins? You don't just stitch one into the membrane, you stitch these 456, and in the right order and in the right location. You need all this to get, just to process something like topoisomerase going through the nuclear pore. I mean, this is not trivial. And so what evolutionists do, they just show these phylogenetic trees and say, oh, it evolved, that's proof. They don't account for the complexity of all the parts that have to be there. And just like Darwin said, if you could show something that couldn't be evolved in numerous slight successive modifications, his theory would fall apart. And I'm like, isn't this good enough? It's good enough for me. It's good enough for a lot of people. So let me now terminate this. So now going back to, let's see where we were. I was talking about the topoisomerase. So we saw the nuclear import and export. We saw everything that the topoisomerase has, but even just getting it in the right place to do its job is also a big deal. And it needs the nuclear localization signal. Let me show you the nuclear localization signal in topoisomerase. It's right here. See that big red stretch here? That's the nuclear localization signal. In fact, it's called a bipartite nuclear localization signal because it's actually two signals. This is complicated. This has to be spelled right. And just because we can build phylogenetic trees doesn't mean this is going to evolve naturally. And, because if you don't get the nuclear localization right, the topoisomerase doesn't locate into the cell nucleus. It doesn't untangle DNA, and the cell dies. There's no natural selection at that point. That's why I call it an undarwinable system. And just for grins, so this is one part of the topoisomerase here, near what we call I call it the right side. It's the proper term is, the end of the paragraph. It's called the Carboxy Terminus. The beginning of the protein's the N-terminus. Oops. And I'm just going to show the other parts. So when the topoisomerase has those scissors operating, it needs energy to do that. It's done with ATP, and you can see the ATP binding sites. It's probably like here, one or two of these red things are the ATP binding sites to power it. It has to be there. It also needs magnesium in this DQ region. The little portions here is where it interacts with the DNA. So when it's starting to act like a scissors, and this is not trivial to build something like this. It's only about 1,500 amino acids long. This is giving a feel for the improbability, the undarwinable quality of this. And let me add even more complications. So so I showed how the scissors here, it has to have the right connection points. And then in addition to that, and this is just a different color scheme of the same sequence, you could see the different parts. And we have people studying this for their entire life. These 1,500 or so amino acids, that has been their life because this is so complicated. And as I said, it seems, the phylogeneticist will say, hey, we can build these phylogenetic trees, where we have human here and like a bacteria here. And they say, oh, well, since we can build a tree, it must have evolved naturally. And I'm like, well, what about that localization signal that's in the eukaryote that's not in the prokaryote? How did that evolve? It can't be by slight successive steps for the reasons pointed out. It's undarwinable for evolution. Oh, there again. Oh, now here's another layer of coding. This is what we call the post-translational modifications. Post-translational modifications are like ornaments on your Christmas tree. So you have the branches, you can put little ornaments. On the protein, you can put you can attach chemical modifications, like you could put phosphorus there, or other chemicals, like methyl groups. The whole suite of them. I don't even, I think you can, like sulfur, they call disulfide bonds or bridges. So we can put all these ornaments and kind of like safety pins to hold it together, so to speak, the disulfide bonds. And what you see here are all those areas where we can modify. We can put the Christmas ornaments. And depending on what we're trying to get the topoisomerase to do, it'll have different sets of ornaments on it. So this is almost like a computer memory, but it also changes the shape and function, slightly modifies the function of the topoisomerase. And it does more than just what we saw in the earlier video. We're only beginning to learn some of the other things it does. So we've heard the term protein moonlighting. It probably does some other things that we're just beginning to learn about. So I showed those we call them post-translational modifications. Think of them as Christmas ornaments. And this is another rendering of it. This one shows, this diagram shows, oh boy, sorry. This diagram shows the post-translational modifications on one section of the topoisomerase. So each of these arrows shows the post-translational modification that's often unique to the human. And this was actually part of a publication I had with Joe DeWeese. You can see the name there. Joe DeWeese is a fellow of the Discovery Institute, and he had published in the prestigious scientific journal Nature on the topoisomerases. So what does this mean? You can take a human topoisomerase, put it in yeast, the yeast will be fine. You can't do the reverse because the yeast is not equipped to handle all these post-translational modifications, or ornaments, shall we say, these Christmas ornaments. That means the human being is way more complicated. Even for the same protein, it is more complicated. So when people try to say it's just, you know, we have a common ancestor and then we just radiate from that, it doesn't necessarily mean complexity. Well, increases. In the case of humans, there's definitely an increase of complexity from the primitive. We have experimental evidence of that because of the post-translational modifications. So let's back up a bit because I've thrown a lot of data at you. We have what may look just like a simple, you know, just like letters of amino acids. But they're layers of overlapping codes and things to keep track of that make this work. So I keep showing the same set of letters, but I'm coloring it differently just to show the different layers of complexity. Same set of letters. These are the post-translational modifications. Earlier I showed the connection points in forming those pair of scissors, what we call a homodimer. So, boy, it's already an hour and a half. I'm going to have to figure out how to cut this down. So you could see that this can't, it's very hard to evolve this from random mutation. There are equivocations that evolutionary biologists use where they say, oh, we're able to evolve some sort of catalytic ability from random peptides, like say 15 amino acids long. And then they'll generalize that. It's an invalid generalization to say, oh, then we can evolve something like topoisomerase. Topoisomerase catalyzes. These random peptides catalyze. Therefore, their logic goes, we can make a topoisomerase since they both catalyze reactions. I hope I've dispelled that notion completely here. That's very misleading and not very honest. Hang on, I had to take care of someone there. So that's topoisomerase. So now, just talking a little bit about probabilities, and this is stuff I've covered before. One way we can establish improbability without appeal to anything metaphysical is using things like the law of large numbers. So here's an example. Let's say we had fair coins. This is illustrating if I just sort of randomly flipped them but did arrange them, you could see it'd be about 50% heads, 50% tails. What would be improbable is that they're all heads. And the reason this would stand out is it's a violation of the law of large numbers. LLN is the abbreviation. There are other things of improbability we could see, like if I just randomly picked out cards and then threw them on the porch, they'd lay out like this. But we would not expect cards to be spontaneously form geometries like this. So again, I'm appealing to, you know, ideas in statistics and physics where we could say something is just not the expected outcome. And that's how we can frame improbability with proteins. and it's not really well developed in my mind yet how we can apply, rigorously the idea of improbability to proteins. Right now, I gave mostly an intuitive argument. Here's an explanation. We see large numbers of violations of the law of large numbers in biology. For example, most of the amino acids in our proteins are left-handed. Most of the sugars are right-handed. The connections are 3', 5'. This is from Change Tan's book. So all this to say, we can calculate improbability. So let me show an example of the law of large numbers. This is an easy example. So collagen's another protein that we express naturally. You can buy collagens supplements or collagens from the beauty store like this, but it is a real protein. It's part of our body. We'd be dead without it. This is the spelling of the alpha-1 paralog of collagen. It's 1,464 amino acids long. Here I'm just expanding the spelling of it. If you want to see a pattern here, I'm going to highlight all the G's, the glycine amino acids, and there you have it. You can clearly see this is a non-random pattern. For it to evolve would have to violate the law of large numbers. I can't get into it here, but you can't invoke any sort of duplication or anything to explain this. It's more complicated, but that's worth a separate discussion. Collagen, then you could say it has this glycine, this motif where you have every third amino acid a glycine, just like that. We can also take the zinc finger protein, human crab zinc finger 136. We can also see a violation of the law of large numbers here. If you didn't have these amino acids in those positions, this would fail. It's very important because the zinc ions have to connect to the C's there. For each row where you have that pattern, and there'll be several zinc ions connected here. That creates the fold. And greetings. Hi, Sal. Enjoying your presentation. Check out the image and animation of the 500 protein bouncers of the nuclear pore complex that selectively let in transcription factors and let out processed mRNA. Whoa. Yeah. Wow. And we have it here. There you go. For those who don't know, Dr. Carter has been a professor of organic and biochemistry. He has done protein engineering on artificial insulins and just a lot of cool stuff. We are very honored to have someone of this caliber join this discussion. And I'm very flattered, Dr. Carter. Thank you for enjoying this and visiting. So, we can group the amino acids in terms of their properties. And if we take that into account, we can even expand. You can see the pattern even strongly here. Okay, no, this is great. Dapper Dino said yesterday that the unusual patterns we see in the protein examples you shared are inevitable to arise through chemical processes. Can you clarify that? Well, since he's not here, we can revisit that. I don't remember him honestly saying that. So I don't want to attribute something to him that he didn't say. So sorry. But let's say someone else said that. Let's say someone else said that. Not Dapper, but let's say it's someone else who said that. Then it's a good question. what do they mean? Do they mean those patterns will arise because it's already in the genome? And I'd say yes. If you have a gene, it's going to eventually arise. But where did this gene come from that has the pattern to define the protein? There's nothing in chemistry that says it has to be one sequence over another. But we definitely know that when we look at mutations. So again, I don't think Dapper would say that, but if someone else did, I'd say, well, that's misleading. Because if you say it naturally arises, it's like, yeah, if the gene is already there, it wouldn't arise naturally if the gene doesn't exist. And I've tried to, topoisomerase is one example. So that is a good question. So let me move on. And greetings here. Thank you. Greetings, Cram. So let's just move on. So we have this motif here, like that, where you have the one in red is the cysteine, cysteine, histidine, histidine, and then some other ones here. That's the structure. What that means, though, is you have, for one, you can't evolve a collagen into a zinc finger or vice versa. And then it's also unreasonable. I mean, you can evolve it in slight successive modifications. I mean, if you try to modify this to look like this in slight successive modifications, you're going to kill its characteristics. It's going to cease to function, vice versa. And they'll say, well, that's not how it happened. It came from a common ancestor. And just like, okay, describe the common ancestor. If you can't describe it, then it's just on faith that you're saying it evolves. So even evolutionary biologists will say they have independent ancestry. And it's like, okay, independent ancestry in the sense you don't have a common ancestor and it evolves by slight successive modifications. It has to be some other mechanism, which they won't specify because I believe it never happened. It had to be just kind of created, put in place when the organism first began. So there's no, if you can think about this, there's no macroevolution from one major form to another, or major forms do not proceed from a common ancestor. So this is like, you have architectures here. You have an architecture for a piano, an architecture for a car, architecture for a helicopter. The recognizable architectures, you can change the architectures, the basic architecture, you can make a variation of it, but you're not going to slowly make a piano that becomes a car, all right? Or a car that you might, I guess maybe with some innovation you can make a car that's a helicopter, but a piano to helicopter would be a pretty tough transition. And so it's kind of, that parallels the problem of making protein families. They're very specialized to their function. So what ends up happening is you end up having independent origins of major protein families. Now, it is true that if you take, like, say, the zinc finger of a human and the zinc finger of a pig, zinc finger 136, that is, they will have the same architecture, even though their sequence is only 50 to 60% similar. And so some people argue, well, that's evidence of common ancestry. I'll say, okay, we'll grant that for that protein. We'll grant it for that protein. And we could do the same, they can build these phylogenetic trees. We could do the same for the collagen. The human is on the left. The zebrafish is on the right. They're about 75% similar. And they'll say that's evidence of common descent. But the problem is you can't make those trees unify to one protein. So even if you assume universal common ancestry in terms of the entire, organisms, the individual proteins had to have separate ancestries because there's no gradual, there's not any gradual transition from one to the other from a common ancestor. Even they generally, even Dr. Dan conceded that, and he gave the same reasons I did. He said, I can't put it through my my alignment process. I can't put the sequences in an aligner and they're going to align because they're different families. It's like comparing apples to oranges. So we have, at least at the protein level, they have unwittingly given us independent origins of major protein families. And I would argue this is kind of the beginning of taking evolutionary theory apart. Universal common ancestry. This is one way to attack it. We can attack it in terms of the evolution of the eukaryotes. So we're actually cutting the phylogenetic tree at the very base. We can also show it at the protein level. If we're able to show different cellular architectures, it'll start to compromise universal common ancestry at higher levels. But I can at least show kind of the idea of macroevolution not happening in gradual steps. It's undarwinable, so to speak, at the protein level. So, Darwin said if you can show something that won't evolve by slight successive steps, his theory would fall apart. And so the reason I'm showing some of this is we could argue that, if it's a collagen and a collagen in humans, collagen in a zebrafish, yeah, slight successive steps will work to explain the differences and the changes. What it will not do, you can't use slight successive steps, though, to form new major protein families. And this is not highlighted in evolutionary textbooks, and it's understandable. So that's a little bit of the law of large numbers. So how does this relate to any papers we're working on? There's Dennis Venema. He wrote Atom and the Genome. And his favorite example, Dennis Venema's favorite, okay, he's a theistic evolutionist. He's trying to say how evolution's so proven. His favorite example of evolution is the nylon-eating bacteria. And then for the Discovery Institute folks, it was also a favorite example by Ken Miller, who was involved in the infamous Dover trial. So here's a picture of Ken Miller was an expert witness there. And here's a picture of Ken Miller boasting about how good evolutionary theory is. So Ken Miller used the example of nylon-eating bacteria in a famous case. The NCSC used this nylon-eating bacteria to argue that we can evolve new proteins without God's help. So actually, Ken Miller didn't use it in this case. He used it in the book in the aftermath of this case. And then Dennis Venema in 2016, he was using it as his favorite example of evolution of new proteins. And he cited the Proceedings of National Academy paper by Ono that talked about birth of this new enzyme that enables bacteria to eat nylon. It turns out that was false. Dr. Sanford and I have released a paper on this. An editor has approached us to publish it. We've had 7 rejections, not because of what we said was wrong. They just said they wouldn't publish it. The editor would say to us, your conclusions are correct, but we're not going to publish. And they gave a variety of lame reasons, in my opinion. Thankfully, the editor of Cell Reports, Steve Matheson, this editor-in-chief, publicly affirmed that our conclusions were correct. Ono was wrong. But that also means that the star witness for the Darwin side in the Kitzmiller versus Dover trial, he was wrong. His example of evolution of a new protein was wrong. And it was just terrible how he got his facts so wrong. It was unbelievable in the book. And we tore him up. And editors didn't like us, telling the truth. Same for Dennis Venema. A little backstory to this. How did we get more confident that we were right? Dr. Sanford sent me to the NIH. My professor, a top-tier evolutionary computational biologist, was my professor. I said, this doesn't look evolved to me. It looked like that enzyme existed before the formation of nylon. He said, absolutely, in his Russian accent. So we're writing papers to criticize things that are still being published in the Proceedings of the National Academy, still being published in Nature, that are wrong. It may take 35 years to correct, but some of this wrong stuff is still cited in the scientific literature as true. And, the current theory, just kind of a rehash of this frameshift mutation hypothesis that, Dennis Venema was so enthusiastic over. You frameshift the genes of any of the proteins I just showed, you're going to blow them apart. I showed the complexity just to show you that it's going to blow it apart. And yet this will get published in a top-tier journal, that this is an effective strategy for making new proteins. It's an effective strategy for making junk, functionless junk, or disabling existing proteins. I can't believe that this would get in the Proceedings of the National Academy of Science, but it's pro-evolution. Not that they ever really tested this on complex multimeric proteins. Oh, by the way, I said multimeric proteins. Those are the proteins that need many copies, just like the topoisomerase needs 2. That potassium ion channel that I showed, that actually is 4 of them, identical copies coming together to make the potassium ion channel. So my final thoughts on this. Oh, Swami Das. Lots of nice comments in the chat. Oh, yes, I didn't know that. Oh, yeah, he does have an attachment to it. And the defenders of that, as I just showed, they're hanging their hat on something that we've shown as a hoax. I mean, it wasn't originally a hoax, but at this stage of the game, it is a hoax. So yeah, I have to say maybe, some people really don't want to publish our paper because it makes the whole industry look bad. I'm just like, well, doesn't truth count for something? And to the editor's credit, they keep saying, well, maybe you could find another journal, good luck. We think you're right, but maybe someone else will publish it, not me. And I do respect that, especially if they're closet ID proponents not wanting to lose their job. So finally, I'm going to just say, after all this, I'm going to just criticize, oh, that's Emily Reeves. That's the wrong Emily Reeves. A few times I was telling Emery, can you just show her picture? Because Emily Reeves is part of the Discovery Institute group that I'm speaking to. There is an Emily Reeves who looks like this. That's not the Discovery Institute Emily Reeves. She's some sort of host for a show about cars, and she repairs cars, and she shows off how she repairs cars. The Emily Reeves that I'll be speaking to tomorrow is a different Emily Reeves. She's a biochemist. So anyway, so I have some final words to say. What I focused on is the complexities of biology. I'm just going to suggest we avoid arguments that invite distraction, obfuscation, misdirection. The other side will often say, you know, ID proponents are tempted to try to say ID is science. And I'll say, don't argue that. ID can be true. It doesn't have to be science to be true. George Washington existed. That's accepted as truth. I wouldn't classify that as a scientific claim, but it's true. So intelligent design I put in the same category. It's true, but I wouldn't classify it as science. Don't get dragged down into debates over whether ID is science or not. Another one, people talk about irreducible complexity being a challenge to Darwinism. And I'm not, you know, I respect Michael Behe, but there's a rhetorical maneuver we have to be very careful of. And I saw Dr. Dan and Dapper Dino use it very effectively. What they'd say is they found another system that's irreducibly complex and that they could demonstrate that it evolved, even though it's irreducibly complex. They were, Dapper Dino was in a debate with Otangelo. And then Otangelo was writing me, he said, Sal, what do I do about this? I said, for one thing, don't debate and say irreducible complexity is a problem for Darwinism. You're going to have too many rhetorical maneuvers. And I knew exactly what was going to happen. They're going to say, we found an irreducibly complex system and it evolved in the lab. And like Otangelo, you see, that's the problem. You don't know about that experiment. You don't know all the details. You're making a claim that it didn't evolve. And it may have, it may not have. Why are you going to, you know, they're going to derail you and try to get you into an argument that you know nothing about. What you should have done is not use the word irreducibly complex. You should pick one system and then show that it is what I call unevolvable, undarwinable, so to speak. And you use Darwin's quote here from The Origin of Species, organs of extreme perfection and complication. Here it is. If it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous slight successive modifications, my theory would absolutely break down. So what am I saying? You only need one system. Don't try to say all irreducibly complex systems will not evolve. They're going to play rhetorical games with you. They're going to claim all sorts of irreducibly. They're going to claim all sorts of systems are irreducibly complex, like the HIV viroporin, and then say, well, we have evidence it evolved. See, if you try to make these sweeping, grand claims across all biology, they're going to get you. And that's what happens when you say irreducible complexity is a problem for Darwin. What I would suggest, instead of using that word, that phrase, use Darwin's own words. If it could be demonstrated that any complex organ, which means if you can demonstrate just one, his theory breaks down. So when Otangelo gets on the channels and say irreducibly complex systems are a problem for Darwin, he's trying to say all these systems, you're going to have to account for all of them, and you really only need to have just one. So don't fall into that trap. The other thing is, yeah, it depends on how you say irreducibly complex. If you're saying all of them are unevolvable, that's indefensible because you'd have to know every system that ever existed. It's just not worth it. Don't fall into that rhetorical trap. And I just see ID proponents getting just pulled into these definitional or arguments over definitions. You never throw complexity on the table as I've tried to do here. When you get to the point you're showing complexity and the unevolvability of individual specific systems, your case is stronger. So let me see here. And I've tried to say we frame biology in terms, improbability. Try not to use the word function, okay? Because if you're trying to prove teleology, if you use the word function, you're starting to use teleological language already. That means you're trying to make your conclusion when your premise is already equivalent to your conclusion. You just have to be very careful. That's why I tried to use the ideas of geometry, connectivity, and the law of large numbers to demonstrate improbability. There are probably more sophisticated ways to do this without invoking the word function. And so you can also specify, you can make quote-unquote requirement specifications, and engineers understand that, without necessarily using teleological language. You can then state the improbability and difficulty of evolving such systems. At that point, it becomes, when you can show that it's improbable, then you can say, you can admit teleology into biology. And so that's all I have for tonight. Thank you for the audience as I try to do a rehearsal of my presentation tomorrow. And I'm going to have to at least cut this maybe by 75%. So this was a good chance to actually just see how quickly I could go through this. So, yeah, thank you. Exactly. How are all these parts and functions going to come together at the same time when science can't even get one of the working parts? Yes. Oh, and thank you. Loss of any information or function is not a mechanism you should expect, or how for in the theory of evolution. If the loss of specified proteins had a net loss of function, you die. At least 8 parts needed to function in the simplest single cells. These 8 or more parts must do a minimum of 10 functions. Well, I never heard those figures before. I've heard figures on the order of 400 proteins needed. 400 proteins needed, at least. So you're generous to say only 8 parts. We need even more. So with that, I look forward to seeing you all again, and thank you very much for your audience. Take care, and God bless you.